दिख रही है? चलो ठीक है। शाम को आ रही है वॉइस? ठीक है। Assalamu alaikum. A very good morning to all of you. This is Almina Khan, the moderator of the day 2. I'm representing here AIRC. I wholeheartedly welcome you all on International Brain Research Organization, IBRO, APRC, Associate School of Neuroscience, theme entitled as Untangling the Degenerating Brains from Neurobiology to Psychopathology. It's a 6-day school virtual program comprised of talks, activities, and lab tutorials from different speakers from all around the globe. I really hope you had great time during the yesterday's sessions. We have an amazing line of speakers for today as well. Moving ahead, we have Professor Dr. Khalid Iqbal. He is unfortunately not present here physically, but let me introduce him before we officially start. So Professor Dr. Khalid Iqbal is Professor and Chairman, Department of Neurochemistry at the New York State Institute for Basic Research and Development of Disabilities. He received his PhD in biochemistry in 1969 from the University of Edinburgh. Dr. Iqbal was the first to describe in 1974 the bulk isolation and protein composition of neurofibrillary tangles, paired helical filaments PHF from Alzheimer's disease brain. These pioneering studies led Dr. Iqbal and Grantley Iqbal to the development of CNTF, peptidergic compounds, and novel therapeutic approach that involves shifting the balance from neurodegeneration to the regeneration of the brain. Dr. Iqbal is the recipient of many prestigious honors and awards, including the Potemkin Prize from Alzheimer's Disease Research from the American Academy of Neurology, and the Zenith Award from the Alzheimer's Association, USA. He founded and chaired the Biennial International Conference on Alzheimer's Disease from 1988 to 2008. In 2007, the Alzheimer's Association USA established a Khalid Iqbal Lifetime Achievement Award for Alzheimer's Disease Research, which is given out annually at the International Conference on Alzheimer's Disease, ICAD. ICAD to a senior established Alzheimer's disease researcher. Dr. Iqbal has authored over 300 scientific papers in prestigious international scientific journals and edited 7 books on research advances in Alzheimer's disease and related neurodegenerative disorders.
Good morning, everybody. I'm Khalid Iqbal, and I'm very happy to participate in this, the IBRO Associate School in Neuroscience. I've been fortunate to that I was able to attend the very first neuroscience summer school, which was held at Panjwani Center quite many years ago, organized by Dr. Tarinam and myself. And the second summer school was organized by Dr. Taqir Ahmed at NUST. And I guess this is the third, the IBRO Associate Summer School. First of all, I want to thank Dr. Sadaf Ahmed and the other co-organizers for inviting me to participate in the summer school. Today, I'm going to talk about Alzheimer's disease. And these on this first PowerPoint are my disclosures. I've been working on Alzheimer's disease for the last almost 50 years. And, but the reason I want to talk about Alzheimer's disease today is not only because this is the area in which I've been working for so many years, but also Alzheimer's disease is a very cruel disease of the brain and of the mind. And so it has enormous toll in modern society. And so today I would to talk about why, what, and how to treat this disease. Alzheimer's disease is an age-associated disease. And with the improvement in modern medicine in the last several decades, so is the increase in the Alzheimer's disease. This is, as shown in this PowerPoint, these are data from the United States as an example, but this is probably true also for Pakistan and other countries in the world, that if you look at it from 1910 to 2020, in 1910, there were only about 4 million individuals age 65 and over, and now this number is almost 50 million, and it keeps continuing in an exponential manner. And at age 65, there is a 3% chance of suffering from Alzheimer's disease, and that risk for Alzheimer's disease doubles every 5 years thereafter. That means the 3% risk at age 65 becomes 6% at age 70 and 12% at age 75 and so on. So at around age 85, there's almost about 50% risk of suffering from Alzheimer's disease. So, I mean, you can imagine what an enormous problem is this disease. In globally, currently there are about 50 million people who are suffering from Alzheimer's disease, and the projections are that these numbers are going to triple by the year 2050. That means only in about another 30 years, these numbers will triple unless we find a treatment to prevent or inhibit the disease. If you look at the old cases of dementia of middle to old age, Alzheimer's disease accounts for the more than 50% of these cases. And then another 15 to 20% are a mixture of Alzheimer's disease and multi-infarct dementia. So if you combine together, Alzheimer's disease accounts for almost about 75% of all cases of dementia of the middle to old age called senile dementia. Alzheimer's disease is a progressive degenerative disease that attacks the brain and results in dementia, impaired memory, thinking, and behavior. The clinical symptoms of the disease are the disease progresses slowly from mild forgetfulness to confusion to dementia. Histopathologically, Alzheimer's disease is marked by marked neurodegeneration, which is associated with numerous amyloid plaques and neurofibrillary tangles of hyperphosphorylated tau. The most important thing in this disease is, if you look at this image, the one on the left is the normal aged brain, and the similar age Alzheimer's disease brain, there's enormous amount of atrophy. There's as much as 200 to 400 grams of loss of brain mass in Alzheimer's disease patient. an average progression of Alzheimer's disease is about 7 to 10 years. So pathologically, this is a section from the hippocampus of Alzheimer's disease brain. They stained with a silver stain with Chosky. It shows the numerous neurons with neurofibrillary tangle, which are all stained dark, and the neurotic senile plaques. These are all dystrophic neurites, and which surround the extracellular amyloid, not so well seen in this slide. Now, the situation in neuron in Alzheimer's disease is very analogous actually to that of the railroad track. I show you here the railroad tracks, and just like the railroad tracks in the neurons, you have these microtubules, which are called the railroad tracks of the neuron. And then you have the microtubule associated protein tau, which are the railroad ties, which keeps these railroad tracks in position so that in a fixed position, so that the locomotor can move on these tracks and communicate the goods from the center to the periphery and periphery to the center. Just like in case of a neuron, it has to, by exoplasmic transport, it has to transport all macromolecules from the cell body to the nerve terminal, and then retrograde transport from nerve terminal back to the cell body. So, but this structure, this cytoskeleton, which is made up of microtubules and other cytoskeletal protein, but the important thing is the role of microtubule-associated protein, and this is that of tau protein. There are others. The tau is the major microtubule-associated protein of a neuron, especially in the axon. And in the dendrites is the microtubule-associated protein or high molecular weight protein NAP2. So when you have these railroad track checked, you can have all this transport as one is very familiar in case of the railroad. So you require for this, the train to move, for this transport, yeah, you have to have these intact functional railroad tracks. And when the railroad tracks become dysfunctional, as in case of Alzheimer's disease, because of the hyperphosphorylation of microtubule-associated protein, so this transport will suffer. And the result will be, as illustrated here in case of railroad tracks, when the train's not moving, you have all these aggregation of masses. That means here, in case of Alzheimer's neuron, you have all these aggregation of the proteins, yeah? Especially the microtubule-associated protein tau, making neurofibrillary tangle. When this situation gets further worse, when there is, because of the damage to the railroad ties, which is analogous to tau protein in case of neuron, all these railroad tracks will break down. And that's what happens in case of the Alzheimer's disease, the neurons with neurofibrillary tangles. You have a breakdown of railroad tracks. And then, as in case of the railroad tracks, there can be more than one causes of this breakdown of the railroad tracks. Okay, it can be because of the dry rot, or it could be termites. It could be the rust, which hold these points of attachment of the railroad ties, or it can be a physical damage, like somebody puts a dynamite in the roadway tracks, and you can break down those things. And similarly, all these situations actually happens in Alzheimer's disease. And that's why what we say is Alzheimer's disease is multifactorial, and it involves several different etiologic mechanisms. Here in this PowerPoint, it's illustrated here, in about 1% of the Alzheimer's disease cases, they are familial, they are pure genetic cases because of certain mutations in these, the transmembrane protein, the amyloid precursor protein, APRC 1, APRC 2. When you have these certain mutations, and this is in an autosomal dominant fashion, those who inherit these mutations will come down with the familial form of Alzheimer's disease. However, most of the cases, what we believe is almost over 99% of the Alzheimer's disease cases, are sporadic form of the disease. So that means they are not because of any one of these mutations. However, there's the apolipoprotein E4 allele is a risk factor, a very marked risk factor for Alzheimer's disease. Those individuals who inherit only one copy of ApoE from their parents or one of their parents, they have 3.5 times higher risk of having Alzheimer's disease. And those individuals who happen to inherit 2 copies of ApoE4, namely both from their mother and father, they have over 10 times higher risk of having Alzheimer's disease. But again, this is a risk factor and it's not a cause of the disease. Then in addition to that, we know a case of a Down syndrome, which is a severe mental retardation condition, which is caused because of the extra copy of chromosome 21, the chromosomal duplication. You have this. These individuals, have severe mental retardation. They are born with it, but when these individuals are in their 4th decade of life, at the top of Down syndrome, they develop Alzheimer's histopathology, namely Alzheimer's disease, and actually at around age 65, whereas in normal population, you have about 3% risk of having Alzheimer's disease. In Down's cases at age 65, they have almost 70% risk of coming down with Alzheimer's disease. Diabetes is another risk factor for Alzheimer's disease. As I said, traumatic brain injury is another risk factor. It is known that there are studies which have shown that a single traumatic brain injury can lead to, in old age, to Alzheimer's disease. There is brain ischemia, can also be a risk factor, and there are environmental factors. Just like as in Guam Parkinsonism dementia, the presence of the alanine, the BMA, which is a risk factor for the Alzheimer's disease type neurodegeneration. But so in all cases, however, what happens is there is a very marked neurodegeneration, which is associated with A beta plaques and the hyperphosphorylated neurofibrillary tangles. And of course, and these are then the presence of these lesions and this neurodegeneration is associated with the dementia syndrome. This is from a study which we did publish in 2005, but we looked at the cerebrospinal fluid, CSF, from many cases of Alzheimer's and control cases for the level of the markers of these lesions, namely tau, beta amyloid, and ubiquitin, we found out that whereas the normal control cases cluster as one cluster shown in green, the Alzheimer cases formed several different clusters, formed the four major and one very small cluster. There is a cluster with the AD, Alzheimer's disease, with Lewy bodies, where you do not see, you see decrease in amyloid beta as compared to the control cases, but no increase in the CSF level of tau. Whereas there one subgroup where you see a very marked increase, almost threefold increase in CSF level of tau, along with decrease in A beta. And then those cases which are carrier of, most of them were carrier of APOE4 allele, they come here in the middle. They also show about twofold increase in tau and decrease in A beta. But then there is a small group of individual Alzheimer cases where in case of decrease in A beta, they show increase in A beta level, just like I should say, no decrease in A beta level, just like the control cases, but the increase in tau protein. So that means one has to be aware of that the heterogeneity of the Alzheimer's disease, that is probably the subgroups of Alzheimer's disease, and these subgroups may have a different characteristics. So this simple slide actually best represents the puzzle and the challenge of the treating Alzheimer's disease. There's marked neurodegeneration. As I've shown you, there's a huge loss of brain mass. Then there is a beta pathology, which you see as amyloid plaques, and tau pathology, which you see as neurofibrillary tangles. So what is not the absolutely very clear is that the neurodegeneration which leads to the A beta and tau pathology, or the A beta and tau pathology also lead to neurodegeneration. And the data from very many labs actually show all of these. That's why we put it in reversible arrows, that the neurodegeneration leads to both amyloid and tau pathology, and in return, the A beta pathology, especially the tau pathology, can lead to neurodegeneration. But what we do know is that most likely both neurofibrillary tangles and amyloid plaques could be a defense mechanism, because both the A beta soluble especially as oligomers, and the tau as hyperphosphorylated tau, which is not yet polymerized into neurofibrillary tangle, they show cytotoxic activities. So the whole issue is, okay, how to treat this thing, what to treat in this triangle. In Alzheimer's disease field has been very much A beta centric for the last many years, so almost for 30 years. So a lot of effort was put thinking that A beta is the one which initiate the disease process, and based on that, the two scientists, Higgins and Hardy, in 1992 put forward amyloid cascade hypothesis, according to which this is the primary lesion which leads to neurofibrillary degeneration, and that leads to neurodegeneration and dementia. Okay. I will come to in a moment. However, that has not so far proved to be true when in human clinical trial. So another situation here is when the Alzheimer's disease brain undergoes neurodegeneration, so it tries to repair itself by initiating neuroregeneration, but it does not have, because of the old age and the disease situation, it does not seem to have the sufficient appropriate neurotrophic activity to push the reaction from right to left. Okay, so it is, so the balance goes into more towards, certainly much more towards neurodegeneration, and hence you end up getting the cognitive impairment. Okay, so this is the same situation. This is a triangle, and this is the neurofibrillary degeneration. And this process is a very slow, progressive process. It takes place, unlike apoptosis, which occurs in a matter of hours. This takes months and years. Because, as I said, according to amyloid cascade hypothesis, this was believed that it is the amyloidosis which is the primary lesion which drives neurofibrillary degeneration and dementia. And however, all the therapeutic attempts based on this hypothesis so far have shown only that you can, especially with the A-beta immunotherapy, you can reduce the amyloid load, namely the plaque amyloids, but it does not seem to affect neurofibrillary degeneration, and also does not help clinically. The patients do not show any rescue of the dementia syndrome. Now, probably all of you must have seen the news just several days ago. The Biogens had a monoclonal antibody, aducanumab, and they got did the human clinical trials, and based on that, although the trial showed us the treatment with this monoclonal antibody to A-beta, which labels both the A-beta oligomers and clears amyloid plaques, but it did not show any clinical therapeutic beneficial effect. Despite that fact, the FDA took very unusual step of approving this treatment conditionally for 9 years, that the Biogen, the pharmaceutical company, will collect the data to see whether the long-term treatment with this A-beta antibody, which clears amyloid plaques from the brain, will show any clinical benefit. Actually, it was so controversial that 3 members of the advisory board of FDA, they resigned in protest from the advisory committee. Okay, so just like the A-beta immunotherapy, there's been also many studies by many labs carrying out both active and passive immunization for tau. In our own lab, we found out that we had antibody to a proximal amino terminal of tau that not only could inhibit tau, but also A-beta pathology in this transgenic mouse model of Alzheimer's disease. This was the first study which showed that actually that by inhibiting tau pathology, you also inhibit the A-beta pathology. So I'm not, because of time, I'm not going to go very much to that, but I just want to let you know that currently, this is a very hot area of research for Alzheimer immunotherapy, and that the tau antibodies by several groups have been found to be safe in phase 1 human clinical trials, and now several phase 2 clinical trials are underway. There's only one phase 2 clinical trial which has been completed. This is by Exxon Neuroscience. This company is based in Slovakia, and the paper that came out last month in Aging, in Nature Aging. You can look it up. Briefly, what they found out is that after the, I think 11 doses of the active vaccine, these Alzheimer's disease individuals, the patients who were younger and had higher level of CSF tau and most recent onset, more aggressive form of the disease, they showed a very significant reduction in the disease biomarkers in the CSF, namely the neurofilament light chain for neurodegeneration, and the tau and phospho-tau levels were decreased. And there was some indication of also clinical benefit in these patients, in their cognitive impairment. I just want to point out here that the tau pathology is not unique to Alzheimer's disease. There's a whole group of family of diseases called tauopathies, where you also see the tau pathology. But the important thing is, irrespective of, whenever you see tau pathology in any disease, any condition, it is always made up of hyperphosphorylated tau. You do not see in vivo any tau aggregation which is not made up of hyperphosphorylated tau. Okay, that's important. Okay, because that's the key to it, and based on that, there are a lot of therapeutic attempts are also being made to see how we can inhibit hyperphosphorylation of tau. Okay, as I said earlier, the Alzheimer's disease brain is a response to neurodegeneration by trying to initiate neuroregeneration. And as you know, the only area of the brain where you can have, because since the neurons are post-mitotic cells, they cannot divide. So the only area of the brain, the mammalian brain, where neurogenesis takes place, that is in the hippocampus, is the subgranular zone and subventricular zone. Okay, and here we looked at it several years ago, as using MAP2, which is a dendritic marker, using antibodies to MAP2, MAP2AB are mature MAP2, and MAP2C is an immature MAP2, which you see in immature nerve cell. So when we look at it, we found, unlike in control brain, there was a very marked reduction in the mature MAP2 in Alzheimer's disease brain, but there was quite significant immunoreactivity for immature MAP2, MAP2C, suggesting that, consistent with thinking that the Alzheimer's disease brain responds to neurodegeneration by trying to initiate the formation, by regenerating neurons or formation of new nerve cell, neurogenesis, but it is not successful because you see only the immature MAP2, you do not see mature MAP2. So with this idea, so we came up with the ideas, so we should not only think for treatment of Alzheimer's disease to inhibit neurodegeneration, but since we know this is the sequence in which the neurodegeneration takes place in Alzheimer's, here's a neuron which loses all its synapses and dendritic arborization. In the end, it is like a tree with just a stump, and the cell neuron dies. Okay, so I mean, this level, although the cell body is still there in the brain, but it has very little function left because it doesn't have any connectivity left. So the idea is, okay, we came up with the ideas. So we should not think only to inhibit this process, but we also think, okay, that will be fine. Okay, if we can stop this neurodegeneration right in the very beginning, at the so-called the zero stage of the disease, but that's not always the possible, because we do not know actually when exactly Alzheimer's disease starts in an individual. Yeah, so we have to think only also if we can. The only way to be able to treat them successfully would be if we can regenerate this cell and get back from the right to left, the regeneration. You could do this process. So with that in mind, we developed this compound. This is a peptidyl compound, PO21, which is based on ciliary neurotrophic factor. And what this compound does is, this is the scheme of neurogenesis, but this compound this compound inhibits the activity of LIF, leukemia inhibitory factor, which in the case of the stem cells of the brain, it keeps them in a transient amplifying cell state. It does not let this process further have them to become neuroprogenitor cell, which is the precursor to neurons. So it stops here. So what this compound does, it inhibits the LIF activity. So this checkpoint, so now the neurogenesis can proceed. And second thing what this compound does is, it increases the activity of the protein kinase A, alpha subunit, which will phosphorylate the CREB, transcription factor CREB, to phospho-CREB, which is the active form of the CREB, and that is the transcription factor which will transcribe BDNF. So you get increased level of BDNF. And second, it also, the LIF binds to CNTF, LIF, GP130 tripartite receptor through the JAK-STAT3 pathway. It also affects transcription of CREB as well as the BDNF. So, but net result is we see increase in the level of BDNF, and that is required for the maturation of these neuroprogenitor cells from immature neurons to mature neurons. Another beneficial effect of this BDNF is, which is the very much increase as a result of the PO21 treatment in vivo, is that now is through binding through TrkB receptor, will activation of PI3 kinase, which will activate AKT and will result in increase in phosphorylation of GSK3 beta at CD9, which is inhibitory, and that means it will prevent hyperphosphorylation of tau to hyperphosphorylation. As you know, GSK3 beta is a major tau protein kinase. Similarly, it also will prevent the inhibition of GSK3, will also prevent the phosphorylation of the amyloid precursor protein at threonine 668, which favors its amyloidogenic processing into A beta. So we've also seen that when this compound is a blood-brain barrier permeable, it's a small compound, which is a 578.3, and it also engages the target JAK-STAT3 pathway. We can show that in the transgenic animals, which will inhibit the phosphorylation of STAT3 as tyrosine 705. So Using these compounds, Faraz Qasim, during his PhD work in a lab, he treated this transgenic mouse model of Alzheimer's disease, which has the APP presenilin 1 mutations, which causes AD, and plus it has the tau P301L mutation, which one sees not in Alzheimer's disease, because Alzheimer's disease does not have any tau mutation, but in frontotemporal dementia. When you put that, you can produce both the A beta and tau pathology in the same mouse. So using this mouse, he treated them when they were 9 to 10 months old for up to 12 months with this compound in their diet. So we found out the chronic treatment with this compound not only rescued neurogenesis and neuronal plasticity deficits, but also reduced tau and A beta pathology. Show you only very few slides from this study here. Just want to show you that in these animals, after 6 months treatment, when they were 50 to 60 months old, you see that in the MAP2 level in CA1, CA3, and indented gyrus. In all cases, this is the control wild type animals. This is transgenic mice. You see very much reduction in MAP2 expression and the almost complete rescue of the MAP2 expression in animals treated with this compound PO21. And these are the quantitations data: reduction and rescue, reduction, rescue, reduction, and rescue. Similarly, in addition to this, when we look at the hyperphosphorylation of tau, we found out both at 6 months treatment and 12 months treatment. That's when animals were 50 to 60 months old or 20 to 21 months old. There's a very marked reduction in tau pathology as seen with this antibody 88, which recognizes hyperphosphorylation of tau at serine 202, threonine 205. This is in CA1 area. This is in subiculum. You see, this is a transgenic mice and transgenic mice which were treated with PO21. There's a very marked reduction in tau pathology, which we see there. And they both at the after 6 month treatment and same thing after the 12 months treatment within animals were 21 to 22 months old. Now, it is also now Nergis Missouri, another PhD student, she treated these animals when they were 3 months old. At 3 months, these animals are already cognitively impaired, but they do not have any tau or A beta pathology at 3 months. So treat them for 3 months onwards for 18 months on this compound orally in the diet. She saw a very marked reduction in A beta pathology as shown here. This transgenic, and this is PO1 treated animals, reduction in the amyloid plaques, and this is the quantitation, and as well as in tau pathology, with 88 antibody, CA1 area and subiculum. So this showed us in both studies, independent studies, that the chronic treatment with this compound, which promotes neurogenesis and neuronal plasticity, not only that you see increase in neurogenesis and the neuroplasticity protein markers, but to see a very marked reduction both in tau and A beta pathologies. This led for us to do another study. So what happens in Down syndrome? As I said earlier, in case of Down syndrome, every one of these individuals, when they are in their 4th decade of life, they get at the top of Down syndrome, they get Alzheimer's histopathology, just like any Alzheimer's disease cases. Lots of the neurofibrillary tangles and amyloid plaques, especially a lot of amyloid plaques, because of the extra copy of amyloid precursor protein, which resides on chromosome 21. yeah, because of trisomy 21. So the idea was, okay, if we treat these animals, the mice, from gestation day 8, because gestation day 8 is roughly corresponds to the start of second trimester. That's when the amniocentesis is done, and one can find out whether the fetus, which is the mother is carrying, is a normal or a Down's fetus. So we tried to mimic that in mice and treated them only for that period. And when the animals are 21 days old, and then you put them, separate them from the mother and put them on a normal diet. So this PO21 was given to the expectant mothers all the time. Okay, so the pups got the PO21 treatment first through the placenta, and when they were born through the mother's milk. And in the last few days, they can also start probably nibbling some of the diet. Okay, so based on that, another postdoc, Ray, he started to look into it. Okay, we know, of course, the Alzheimer's disease is different from Down syndrome, but even in Alzheimer's disease, in case of familial Alzheimer's disease cases, of course, just like in Down syndrome, these individuals are born with those disease-carrying mutations in APP, presenilin, or 1 or presenilin 2. So that means they could also have some abnormalities during development, which may account for them to get Alzheimer's disease later in life. So with that in mind, so Wei did the replicated for us the experimental design as shown here, but using the triple transgenic mice in case of the trisomic mouse model of Down syndrome. So when he did that, but he found out that, first of all, let me show you here the behavioral data. This is on Y-maze. These are the open arms entries. The transgenic mice were impaired, showed the impairment. There were a reduction in the entries, but not in the PO1 treated animals. Same thing in the time spent in open arms and the distance traveled in open arms. Only the transgenic mice showed reduction, but not those transgenic mice which were treated with the compound. Then when looking at the novel object recognition test, this is the sample phase. They spend equal time with object 1 and object 2. And in the test phase, in discrimination index, the transgenic animals show very clear impairment, very marked reduction in discrimination index, which was completely rescued in those animals which were treated with PO21. In mouse water maze, in the escape latency, similarly, these are the transgenic animals in red, and the blue is when they were treated with the PO21. You see the significant decrease in escape latency in the mouse water maze. This is the first entrance into the target quadrant. The latency, the transgenic animals took much longer time to first enter to the target quadrant, whereas the transgenic animal treated with PO21 did not. Similarly here, percent time in target quadrant was reduced in transgenic mice, but not in animals treated with PO21. This is these, and they all have the same swim speed. Okay, so when we look at the data on the tau pathology, there's a Western blots and quantitation. I just want to point out the main thing is that there was always compare the red with the blue one. The black and green one are the wild type animals with and without and with treatment, and red as the without and blue is with treatment. You see the R4130 134D and 43D, these are the antibody recognizing tau. 134D recognize total tau, 43D only human tau, the transgenic tau. You see the reduction in it. And when you look at the hyperphosphorylation of tau, there was a reduction both at the 4 months after treatment and after after about 18 months after treatment, as shown here. Yeah, actually 22 months after treatment. Sorry, 22 months after treatment. And this is the quantitation, the histochemical data. Again, this is the untreated transgenic mice, and it's a treated one. You see the very marked reduction in tau pathology in these animals. And the important thing is, these animals were treated with this compound PO21 only from gestation day 8 to postnatal day 21. And you see its effect or beneficial effect all the way into the old age at the age 22, at 22 months of age. Okay, so this is very analogous sort of the situation in Alzheimer's disease. Yeah, here again, looking at in this study for amyloid pathology, this is in the show you the triple transgenic animals, A beta pathology, and this is in the pure 2-1 treated animals, which is very marked reduction. And this is in these corresponding area. This is in the subiculum, the transgenic, untreated, treated. This is in piriform layer, untreated, treated. This is in the cortex, the forebrain. This is untreated, treated. This is in the hippocampus, untreated, treated. In all cases, you see reduction in amyloid pathology. This is also shown here. The plaque load, the untreated, treated animals. This is the high MAC, untreated, and the treated animals. And quantitation show a very marked reduction in the A beta plaque load in the treated animals as compared to untreated animals. This is the with A beta immunostaining, and this is with thioflumin S, the similar picture. And also here we look at the phosphorylation of APP at threonine 668. Again, the transgenic animals shows increase as compared to the wild type animals, and treated animals shows the marked reduction completely as the normal animals in phosphorylation of APP at threonine 668. Here, then looking at it, the important thing here is looking all the neuroplasticity markers. But I want to point out most importantly here is both in the 4 months old and at 22 months old animals, you see a very marked increase in the CREB activity. Phospho-CREB over CREB ratio is very much increased in both cases. Finally, look at also neuroinflammation markers. We did not see any significant change in the microglia, but we saw a very marked reduction in astrogliosis in the animals treated with PO21 as compared to untreated animals. They both at 4 months and at 22 months of age, as shown here. The untreated, treated, the untreated and treated animals. So what it looks like is, in summary, that the unlike the A beta immunotherapy, tau immunotherapy, I'm sorry because of time did not show all the data to you but there we had found that it reduces both the tau pathologies and amyloidosis, and you see the reduction in cognitive impairment. But most interestingly is that by using neurotrophic support, which reverses the balance from neurodegeneration to neuroregeneration, we see reduction of both amyloidosis and tau pathology of every degeneration, and we see the reversal in this case we saw of the cognitive impairment. So we think we can this inhibits not only this process but this the reverses process from right to left here. Okay, that's shown here. This the neurotrophic support. All right, with that, I thank you all. And these are the, actually not all, some of the people whose data I showed you today, who worked in my lab, and formerly when Inga was alive in her lab, the Dr. Felu's lab, and Gong's lab, and Chungling Dai and Benhu from our group, research group. I'm sorry, it's because of time difference, it's going to be very different. The timing between in Karachi and in New York, we are 10 hours time difference. So when this presentation will be made, it will be quite late for me. It'll be almost midnight. But I'll be very happy to, if you can email me all your questions, I'll be very happy to answer them through email, or we can have an appropriate time, another Zoom meeting. Okay, with that, thank you all. Bye.
Thank you very much, Professor Dr. Khalid Iqbal, for your valuable time. We greatly enjoyed your talk. Due to the time acquaintance, Dr. Khalid Iqbal wasn't able to make it. But you all can post your queries and questions related to his talk in the comment section or in the chat box. We will get back to you once he responds us. Moving on, we have our next speaker from Karachi, Pakistan, Professor Dr. Daraksha. Professor Dr. Daraksha has acquired her PhD degree in neurochemistry from University of London. She also has an honorary degree from University of Complementary Medicine, Colombo. She is working as Emeritus Professor at Dr. Panjwani Center for Molecular Medicine and Drug Research, ICCBS, University of Karachi. She has served her period as the Dean Faculty of Sciences and Chairperson of Department of Biochemistry, University of Karachi. She has over 250 research publications in international and national journals of neurochemistry, neuropharmacology, biochemistry, and other life sciences. She has done a spectacular job and has supervised over 40 PhD and MPhil students and over 40 MSc thesis students. She has received multiple grants for research projects. She is a lifetime member of many scientific organizations and societies, including Pakistan Society of Biochemists, Pakistan Pharmacological Society, ISBPP, which stands for International Study Group for Biochemical and Biophysical Research in Pakistan, and IBRO, International Research Organization. She was also awarded multiple times for her extensive services as a mentor and researcher. Unfortunately, Professor Dr. Daraksha is not available right now with us due to uncertain health situation. We really wish ma'am a speedy recovery, and we will definitely entertain her in the next sessions. Moving ahead, our next speaker for the day is Professor Dr. Zaman Khan from Allama Iqbal Medical College, Lahore, Pakistan. I would like you to introduce with the introduction of Professor Dr. Zaman. Professor Dr. Zaman has completed his FCPS in internal medicine from College of Physicians and Surgeons, Pakistan. He did his postgraduate residency in the field of internal medicine, diagnosis, and treatment of medical illness. He has a very vast experience in research, most certainly in infectious diseases. He is serving as an assistant professor at Lama Iqbal Medical College. He is a member of Dengue Export Advisory Group Punjab, and he is an active focal person for dengue fever at Jinnah Department at Jinnah Hospital, Lahore, Pakistan. Also, he is a demonstrator at Department of Physiology, Central Park Medical College, Lahore, Pakistan. He has gained experience in cardiology, internal medicine, ICU, and EFT. We welcome you on board with us, Professor Dr. Zaman. Over to you.
Thank you, Zarmeena, for this kind introduction. I hope you can hear me.
Yes, we can. Sure.
Okay, thank you. And I mean, just since I'm delivering this lecture live, so if there is any interruption during the lecture, just let me know. Yeah, so like I can switch to another network. So as I have been kindly introduced by Zarmeena, I am Dr. Mohammed Zaman. My interest is primarily in neurogenetic diseases, and primarily in neurodevelopmental disorders. So we have been working on elucidating the molecular basis of intellectual disability. But today I am going to talk about a very interesting topic. It's my understanding that we have participants from diverse backgrounds with different educational backgrounds. So I have decided to keep this lecture more generic so that everybody can understand about this, the new advancements in the field of neuroscience. So I'm going to share my screen now. So I hope everybody can see that. Zarmeena, can you see my screen?
Yes, sir, we can see it.
Okay, right. Thank you. So, so the title of my today's talk is Brain Organoids, from modeling neurodevelopmental disorders to studying neurodegeneration. As I have been mentioned, I am an assistant professor of medicine with special interest in the field of neurogenetics. Well, you are all aware that there are a number of animal models that have been used to study neurological disorders, including zebrafish, flies, Drosophila, rodent models, rat and mouse models have been used. And then recently, non-human primates have been used to model neurological disorders. Well, every model has their own pros and cons. So I think rodent model is commonly used to study since the advent of transgenic mice technology. I think they have extensively been used in neuroscience. And one of the benefit of using these rodent models is that they mimic many characteristics of human brain, and also it's easy to study behavior of mice in the lab. So we can look at various structure and functional abnormalities and then compare them with the behavioral abnormalities at the animal model. I think a lot of genetic insight has come from studies on fruit fly, as well as on the C. elegans. But it has been noted that there are fundamental differences in the rodents as well as mammals. So that has led to the need for studying other primate models, non-human primate models. So monkeys, chimpanzees, they have been used. They are being used in the neuroscience research. The problem is that it's uh, it's not an easy task to actually study non-human primates, and so every model has their own pros and cons. So that one of the important limitations of using these animal models is that they usually do not fully recapitulate the human phenotype, human specific phenotype. So there has always been a need uh, to to have models, human specific models that can fully recapitulate the human specific phenotype of a number of diseases. So I'm showing here the brains of different organisms um, for a comparison. As you're aware that human brain has a number of neurons. So for example, elephant brain uh, has twice the weight as compared to the human brain, but that doesn't mean that larger brain doesn't mean larger number of neurons. As a matter of fact, humans have like 4 times higher number of neurons as compared to elephant brain. But then if you look at, like, if you compare the human brain with the mouse brain here, I mean, you can just look at the difference in size. And it's not only the difference in the size, but there are some other characteristics of humans from rodents. So I have magnified the image of mouse brain, and as you can see here, the surface, if you look at the surface of the mouse brain, it's smooth. It's called lissencephalic as compared to human brain that has so many folds and gyri and sulci. So the latest studies have shown that there are other fundamental differences in the development of rodent brains and human brains. So, for example, recently we have seen that there are some distinct populations of cells in human brain that are not mouse model. So here is the mouse developing brain at embryonic day 15.5. So you can see that here is the ventricle, and there is this ventricular zone, subventricular zone, and then there are like cortical plate and other uh, parts of the developing brain. And we can see that there are distinct cell types. So these, like in light green, are the ventricular radial glial cells that give rise to the intermediate progenitor cells that can be seen in gray. And then they further divide and differentiate into mature neurons that then migrate in a radial direction towards the surface of the cortex. At gestational week 10, perhaps the population of these cells in humans looks similar to that in the rodent model. So it's like same set of populations. But later on, what we see is a distinct population of cells. So here in this subventricular zone, we see a new population of cells shown in red, cells. And these outer radial glial cells, they are quite abundant in a zone that now we call as outer subventricular zone. And these, and it is considered that it's because of these large number of outer subventricular zone, outer radial glial cells, or we can call them ORGs, um, these cells have, they lead to the large number of neuron seen in human. So given these differences, these fundamental differences between the mouse brain and human brain, there was a need for human specific modeling of the diseases. A number of other studies have also shown that there are definitely the difference in the population of these cells, outer radial glial cells, between humans and non-human primates. So although they have these outer subventricular zone radial glial cells, the population is less, and that's one of the reasons we see the differences in the cerebral size and the number of neurons seen between human and primates. Human specific modeling of the diseases in recent past, I think in last 2 decades, there have been significant progress in the development of organoid research. So organoids, or we can call them mini organs or organs in a petri dish. So they are the structures that show a number of similarities with the primary organ, and they have the ability to self-organize and self-pattern. So here, as I said, in recent past, organoids have been developed from the pluripotent stem cells, be it embryonic stem cells or iPSCs, and they have been differentiated into various organ tissues. So now we have heart organoids, gut organoids, kidney organoids, liver, prostate, brain, lung, and pancreatic organoids. So, for example, during this pandemic, and I think that's one of the reasons why we are having this IBRO associate school virtually this year, is because of this COVID-19. So lung organoids are being used to study COVID-19, but as we know that the SARS-CoV-2 has effects on various organs, not only the lung organoids, but also brain organoids and kidney organoids are being used to study the effects of SARS-CoV-2. So if we look at the number of papers published in like past 1.5 decade, you can actually see that increasingly more and more papers are being published on organoid research, including the cerebral organoids, retinal organoids, intestinal organoids, and liver organoids. So well, what is the benefit of using organoids? And I will be, for the sake of this lecture, I will be focusing on brain organoids. So what are the benefits of using brain organoids? So if we compare it with the human brain, we can study the temporal order of neurogenesis. So a number of studies have shown that brain organoids, they show the developmental pattern of a normal human brain, so they can actually be used to mimic the brain development in vitro. And then we can see the cellular diversity in the brain organoids that's seen in human brain. And then also with the recent the neurons that are generated in brain organoids, they show at least some feature of functional neurons, and now we have seen that they can be used to they are able to make synapses, and they are able to integrate in various microcircuitry within these brain organoids. But then there are some features that are there in human brain, but not in the brain organoids. For example, the human brain has highly organized structure. So like we see this topology where there are different lobes of the brain. Neurons are organized in a layered pattern. They're very much organized in the human brain, but that's not the case in the current brain organoid protocols. Human brain has complex neural circuits, and we see that the classical gyrification. So some extent of the folding can be seen in brain organoids, but that characteristic gyrification is not seen in human brain. So there are other limitations as well with the brain organoids. So there is like lack of microenvironment. Most of the brain organoids, they lack the blood vessels, capillaries, also the immune cells of the brain, the microglia. We know that they are not derived from neuroepithelium. They're actually mesenchymal in origin. So brain organoids, they do not contain these microglia. So we cannot really, by using these standard protocols, we cannot really study the microglia population and the interaction between the brain neurons and microglia. So, so before we look at how the brain organoid technology came into being, we need to understand. Now we know that there is a need for human-specific model brain organoids because of the fundamental differences between humans and other species that we have discussed. But what are the basic principles that govern the development of these brain organoids? So we have to look back at the development of human brain. And as you're aware that the brain originates, the neural tube originates from the neuroectoderm. And there are a number of transcription factors and growth factors that mediate the development of this neural tube. So as you're aware that they all start as stem cells, but then they differentiate into different lineages, like ectoderm, mesoderm, and endoderm, and then there is neuroectoderm that develops brain. So if you look at this first diagram, you can see that the Wnt signaling and transforming growth factor beta, or fibroblast growth factor, uh, they they are they are present in a gradient. So the lesser amount of Wnt and TGF beta means that these cells will differentiate into ectodermal identity, or more of like neuroectodermal identity. And then these black curved lines also show the gradients of other um, important factors that are there during the normal brain development. So similarly, it's not only like in this cross section, but also there is this craniocaudal and dorsoventral gradient of different transcription factors. So as you can see, there is an increasing craniocaudal gradient of Wnt signaling, um, and if you look at the dorsoventral gradient, we see that higher signaling in the dorsal part is determined by the higher amount of BMP, while the sonic hedgehog signaling is more towards the ventral side. Now, these are the important principles. Like if we have to model the brain development in a petri dish, we have to provide the certain conditions and the amount of the various these transcription factors, so that these iPSCs grow into the neuroectoderm, and then they have the ability to self-organize and self-pattern pattern into these brain-looking structures. So the typical protocol of development of brain organoids include TGF beta inhibitors and BMP inhibitors for the dorsal identity, and then Wnt inhibitors. So the iPSCs develop into embryoid bodies, and then these the next step in vitro is the neural induction, where there is formation of these ventricle-like, cavities, and then they expand, and these embryoid bodies are actually then embedded into matrigel, and then they go on to develop into the embryoid bodies. So the first protocol for the development of brain organoids was established by Madeline Lancaster and Knoblich. And what they did was, and this is the first protocol for the development of brain organoids. There are other groups that have shown the development of retinal organoids earlier, before the Knoblich and Lancaster group developed the protocol for brain organoids. So this is a typical protocol. The human pluripotent stem cells, they form the embryoid bodies. At day 6, neural induction media is added that lead to the formation of this neuroectodermal tissue, which is suspended into the matrigel droplets. So they allow the further three-dimensional growth of these cells, and these are placed initially. They were placed in a spinning bioreactor for development into the cerebral tissue, but now as spinning bioreactors require a lot of amount of the growth media, so they then orbital shakers are now being increasingly used to for this purpose. So then they looked at the structure of these brain organoids. So as you can see in this first protocol, they the brain tissue has neuroectoderm, has self-organized into this brain tissue. So you can see actually tissues with various identities, the tissues that originate from neuroectoderm. So if you look at the far right, you can see that here you can see ventricle-like structure, which is surrounded by SOX2 positive neural progenitor cells. They differentiate into, so this part contains these apical radial glia cells, outer radial glial cells, and then there are intermediate progenitor cells. And they differentiate into the neurons. So like this TUJ1 is a marker, is a neuronal marker. But they, across various organoids, these organoids were highly heterogeneous. So they showed some of the forebrain structures, some so dorsal forebrain structures, and then ventral forebrain structures, and then some of the cerebellar tissue, some of the like retina, retinal epithelial structures were seen in these brain organoids. So, okay, just give me one moment. Okay, right. So, If you look at the three models to study the brain disease, these are the in vitro 2D cell cultures, where we can use single cell lineages or multiple cell lineages and study their structure, versus the study of the cerebral organoids, versus the study of the primary tissue. We all know that the access to the primary tissue is very difficult. So I mean, if you look at the complexity, there is increasing complexity towards the in vivo models, but there is more homogeneity in vitro models, and some medium homogeneity and complexity in the brain organoid tissue. If you further look at the brain organoid tissue, we see some ventricular zone and subventricular zone like areas. We see the various distinct populations of neuroepithelial cells, radial glial cells, outer radial glial cells, intermediate progenitors, and different neurons. But they are not in the this classical, well-defined, layered hierarchy of neurons as is seen in adult brain. So okay, right. So now, I mean, that was the initial protocol, but over time we have further defined protocols. So for example, this unguided protocol that we talked about, developed initially by Lancaster and Noblick, that primarily relies on intrinsic signaling pathways, and they develop cerebral organoids that show different structures. The problem is they are highly heterogeneous, and it becomes very difficult. We do not have uniform of these brain organoids, and they have distinct identity. Compare one brain organoid with another one. But over time, new protocols, that these are guided protocols that rely on extrinsic signaling, have been used to further develop a brain region specific organoids. So now we have like 4 brain organoids, and then amongst these 4 brain organoids, we know that we have dorsal brain organoids, what we call as cortical organoids as well. We have ventral forebrain organoids that have the identity of the ventral tissue. That relies on sonic hedgehog signaling. We have now thalamic organoids. Then there is midbrain and hindbrain organoids. So like we have cerebellar organoids now. So it's very interesting that now we have these region-specific organoids, and one can really focus more on a region-specific organoid to study the disease of interest in that particular area. So in order to study like intellectual disability, now more and more people have relied on these dorsal forebrain organoids. But then, I mean, another problem has been that in reality, when human brain develops, different regions of the brain are connected. So how do we overcome that problem? So like what you're looking at is human brain, human embryo at gestational week 13. And so this is the dorsal forebrain, and this is the ventral forebrain. And this darker colored structure is actually ganglionic eminence. It's medial ganglionic eminence and dorsal ganglionic eminence. Now we know that this thin, dark-looking area surrounding the ventricle is a ventricular zone that is rich in these neural stem cells, neuroepithelial cells. We know that the these neuroepithelial cells and then radial glial cells and intermediate progenitors, they give rise to uh, the neuronal production, and then these neurons move radially and move towards the surface to form different layers of neurons. So this is called radial migration. But then there are neurons that are generated in these ganglionic eminences, and then they travel in a tangential manner. So these are primarily these GABAergic neurons that originate from the ganglionic eminence, primarily the medial ganglionic eminences, and then they migrate in a tangential direction. So the question has been that, for example, these pure dorsal brain organoid, they lack this population of GABAergic neurons that are generated in the ventral part of the brain. So that led to the idea of moving from brain organoids to assembloids. So just again, looking at the neurons and the cells in the dorsal and ventral part of the brain. So they have like distinct identities. The cells in the ventral, in the dorsal part of the brain, forebrain, they show markers like PAC6, TBR1, CTIP2, and SATB2. Cells in the ventral forebrain, they have other markers. So for example, cells from the medial ganglionic eminence, they show positivity for NKX 2.1. Now, it's important to keep that in mind, because these markers are used to really identify these distinct cell populations and then study the migration and interaction of these cells. So so I think around the same time at the Sergio Pasca lab at Stanford, and then an Oblique lab in Austria, they proposed and they demonstrated that what they could do by using specific culture conditionings, they were able to develop these dorsal forebrain organoids and ventral forebrain organoids. So like this human cortical spheroids, it's an abbreviation for dorsal forebrain. Organoid or spheroids have been used interchangeably. And then these are the human subpallial spheroids that show the ventral forebrain structures. So these dorsal forebrain spheroids, they show the markers of dorsal forebrain, including PAC6, CTIP2, and SATB2. So like you can see an abundant population of PAC6 in the dorsal forebrain organoids, but they are negative for NKX 2.1, which is actually a marker of the ventral forebrain. And you can see here in these ventral forebrain organoids that there is a rich population of NKX 2.1 positive neurons. Well, as you can see, like these neurons, they migrate from this area into the dorsal forebrain. So what they observed, a very interesting thing, that when these dorsal and ventral forebrain organoids, they were placed in proximity, they had this ability to fuse. So like macroscopically, you could see that these two structures are fusing together and making one integrated structure that we now call as assembloid. And here you can actually see that. So by using this technique, which is iDisco, we can make these organoids transparent, and then the cells that are in the human subpallial spheroid, which is actually the ventral forebrain organoid, expressing the GFP, EGFP, and you can see that this is the junction between this ventral and dorsal forebrain organoid. And what they were able to see was that actually these neurons, these GFP expressing cells, they migrated from this ventral forebrain organoid into the dorsal forebrain organoid. I mean, they went on to show that actually not only that these GABAergic neurons, they migrate from this ventral to dorsal forebrain organoid, but they were also able to make synapses with the neurons in the dorsal forebrain organoid, and they were able to form these microcircuitry. So, I mean, these brain organoids and these assembloids, in a way, can be really useful to study the interaction between the organoids derived from different brain region specific, structures. And now this technology, I mean, looking again at what you can see here, and I'm going to play this video, Pascal group in Nature in 2017. This is the boundary between the ventral and dorsal forebrain organoids, or the subpallial and cortical spheroids. And the cells, the bright cells here, are actually the GFP expressing cells, and we can play. We can see actually that these cells are. You can see these little yellow arrowheads, that these cells are migrating into the dorsal forebrain. And then they actually use this brain assembloid model to study Timothy syndrome, where they were able to observe that these GABAergic neurons, the upper panel shows the brain organoids derived from the control healthy individuals, and the lower panel is showing the neurons in the Timothy syndrome. And like, if you look at so you can see that as compared to the control, this movement is reduced in cells in Timothy. That not only that this was the Pascal group, but also the Noblick lab. They also showed they were able to actually fuse these ventral and dorsal brain organoids into the assembloids, and here you can see the cells, the green appearing cells from the ventral forebrain entering into the dorsal forebrain organoid. And again, here you can see that these GFP positive cells are moving into the dorsal forebrain organoid. Now that's one and the initial model that used the dorsal and ventral forebrain organoids, and then they combined them into the assembloids, and they were able to study their interaction. Now we have with the availability of the cortical organoids and thalamic organoids, we can combine them into assembloids. Now some labs are working on developing the cortical organoid, and then spinal organoid, and then a muscle organoid, and they are looking at the interaction by assembling these three organoids into one assembloid and study the interaction. And I hope with the every passing day as you can see that there's a lot of interest in this brain organoid technology, and people are working on improving these models, bringing more reproducibility and uniformity in these methods. So, well, the brain organoids, as I said, they were initially designed to study neural development, used extensively in microcephaly research and research on the understanding the genetic basis of intellectual disability, autism spectrum disorder, macrocephaly, lesioncephaly, um, schizophrenia. So, so for example, one of the common observations that have been seen in microcephaly, uh, the brain organoids that are derived from the IPSs derived from patients. Actually, patient-derived IPSs are used to develop brain organoids, and they are compared with the control healthy individuals. What they have seen, for example, one of the differences are that microcephaly brain organoids, they have the less number of early depletion of the progenitor cells, early neuronal differentiation, which means that there would be less number of neurons eventually in the brains of these individuals or in these brain organoids. So that explains the microcephaly phenotype. So that brings us to the neurodegenerative diseases and how the brain organoid technology is being extensively used now in neurodegenerative disease. I would say that use of brain organoids to model the neurodegeneration is relatively newer idea. Like in past few years, people have been using these brain organoids to model um, Alzheimer's disease, Parkinsonism, Lewy body dementia, frontotemporal dementia, etc., the other forms of neurodegenerative diseases. And as you are aware that I will just give one example of use of brain organoids to study Alzheimer's disease, and there are actually a couple of papers out there, and you can go and read more about the use of brain organoids in these neurodegenerative diseases. But before I go on discussing these the use of brain organoids in the modeling of Alzheimer's disease, let's look at the various models that are being used to study Alzheimer's disease. So there are these humanized animal models. As you are all aware that it's not easy to get the phenotype of Alzheimer's disease in animal model. So they are, and these animal models are humanized by overexpression or knock-in of human gene, like APP-PS1 carrying familial mutations are used to get a humanized animal model. Well, what we do, the benefit of using humanized animal model is that they reproduce A beta pathology, so we can see that plaques and oligomers in these brains of these animal models. The brain has microglial cells, so we can see, actually look at the immune reaction there, and we can study the behavior of the model, and we can look at the cognitive impairment. At the mouse does not show these neurofibrillary tangles or the tau pathologies unless we express the tau or their tau mutations. And also, as you know, the mouse develops early, and there is like overexpression of these genes during the early age. So that does not really show the effects of advanced age in the mouse model. That is one of the important risk factors for example, Alzheimer's disease. And also we know that the mouse does not develop the Alzheimer's disease per se. So there is no sporadic pathology. So that's why the many people focused on 2D iPSC's neuron cultures. So the iPSCs are derived from sporadic Alzheimer's disease patients or familial Alzheimer's disease patients, and then they are studied in 2D cultures. So they can, so the pros are that they show features of amyloid beta and tau pathology. It's a genuine human specific, genetic background on which one is working. And one can look at the sporadic case, phenotype case by case across different IPSs derived from different patients of Alzheimer's disease. But then there are some limitations with the 2D culture condition as well. So in 2D, there are no plaques or tangles. There are limited cell interactions. The normally human brain has complex environment that is not seen in 2D cultures. And we know that there is altered transcriptomic profiling of these cells. And many 2D cultures, they lack the immune cells. But now, like, people are using trilineage cell cultures where they are using astrocytes and neurons and then microglia. The problem differentiation of iPSs into there are improved protocol iPSs. And that brings us to the 3D iPSs organoids that now we have. They can be derived again from the Alzheimer's disease patients, be it the sporadic or familial form. They show this complete amyloid beta and tau pathology with the evidence of neuronal degeneration and decrease in the number of neuronal population. We can actually study the complex cell interactions. Well, one of the problem with the brain organoids is that the brain organoid, they show the prenatal transcriptomic profile that it's they show the features of developing human brain. There are no microglial cells in the brain organoids. So now there are protocols that where one can develop the brain organoids, and then one can also differentiate iPSs into microglial cells, and then both can be combined, and we can study the interplay of the microglia and the cells derived from brain organoids. So this paper published by one group in Nature Communication recently made use of the brain organoids. They were developed from the iPSs that were derived from the fibroblast or the peripheral derived bone blood mononuclear cells. So these iPSs, they were derived from control as well as Alzheimer's disease patients. But then amongst control and Alzheimer's disease population, they looked at the APOE4 profile. So APOE profile with E3, or APO with E4 genotype, because APOE4 is one of the very important risk factor for the development of Alzheimer's disease. So now they had 4 groups, and they developed iPSs from these cells, and then by using the protocol for the development of brain organoids, they were able to develop the brain organoids from these patients. And as you can see that, for example, at week 4, you can see this, if you look at this brain organoid, you can see that there are these ventricle-like structures that are surrounded by these SOX2 positive cells. And then there are these green one are the neuronal cells that are seen here. And then if you look at the other markers, for example, these are CTIP2 is the neuronal markers, and you can see like the a distinct architecture where there are SOX2 positive cells are closer to the ventricular surface, and these CTIP2 cells are more towards the periphery of these brain organoid, these structures. And then at week 12, you can see also the SATB2 positive and CTIP2 positive neurons. And then okay, just give me one moment, please. Okay, right. Um, so then they looked at uh, the evidence of apoptosis in these cells. So here is, these are the TUJ1 positive cells in the 4 subgroups of in this study. So these are the healthy controls, and the right ones are the Alzheimer's disease patients. And then you can look at the expression of a cleaved caspase 3, this is immunostaining for cleaved caspase 3. You can see that it's overall increased in patients with Alzheimer's disease as compared to controls. And amongst Alzheimer's disease, and I'm not showing here the quantification, but in the quantification, one could see that the levels of this cleaved caspase 3 are high in Alzheimer's disease patients with APOE4 E4 genotype. These brain organoids, they also displayed the amyloid plaques, amyloid beta deposition could be seen. And also, as you can see, the staining for 88 that actually stains the phosphorylated tau protein. You could see that there are you can see the phosphorylated tau in Alzheimer's disease patients as well as in the controls with the E4 as compared to E3 genotype. So it's what they further did in this study was that they actually looked at the transcriptomic profile, and they compared the transcriptomic profile between the Alzheimer's disease patients and controls and across these genotypes. What they were able to identify that the alteration in the transcriptomic profile was seen in genes that are concerned with the RNA metabolism, RNA synthesis and processing. I mean, overall, this study shows the potential of using brain organoids to model the Alzheimer's disease and other neurodegenerative diseases. So to summarize, brain organoids is a promising technology that have been used to study the brain evolution. So like using brain organoids from a different organism, from the mouse cells and from the non-human primates and human primates, we can actually look at the differences, how brain has evolved across these species and how the brain development is different across these organisms. Brain organoid can be used to look at the tissue architecture, as we see that they mimic the structure of the adult brain, although not fully recapitulate that that structure, but then they mimic, and we can see the distinct cell populations in a layered manner. We can look at the transcriptomic profile by the use of more homogenized models with the greater reproducibility. We can actually compare the transcriptomic profile across various brain organoids. And these brain organoids, they show these transcriptomic signatures that are similar to the primary brain tissue now. And then brain organoids can be used to model various diseases. So we can rely on the patient-derived iPSCs or we can use the iPSCs or embryonic stem cells, and by the use of gene editing CRISPR-Cas9 system, we can introduce various mutations, and then we can study, we can do the disease modeling. I think one of the benefit of these brain organoids is also the ability to do the large throughput screening. So like by using CRISPR-linked technology by the Noblick lab, they were able to, pool cells with different mutations and then grow brain organoid. And then they had this idea of using these barcodes. By using different barcodes, you can actually trace the lineage of different cells and then look at the effects of various mutations. That can be really cost effective, time effective, and that can be used to screen a large number of mutations. Another benefit is the drug screening. By using the brain organoids, you're aware that a number of the drugs that have been shown very promising in the animal model that reduce the plaque size, they, or even in the non-human primates, they were not very successful in the clinical trials. That means there is a need to a human-specific model. So brain organoids can actually be used for a large-scale drug screening. We can see that which drug is effective in preventing the plaque formation or the formation of phosphorylated tau in Alzheimer's disease patients. Brain organoids can, they can play a very important role in the personalized medicine as well, where an individual specific organoids can tell us that which drug is going to be effective in what particular patient. Then we have these brain region specific organoids, and then they can be used as assembloids. And also like electrophysiological studies can be done on the neurons that are derived from these brain organoids. Well, there is a lot that can be, that could be talked about, but I thought that I would keep it simple, and I wanted to give you an overall view of the brain organoid. So that's all. So if there are any questions, I am really happy to take those questions.
Thank you so very much, Professor Dr. Mohammad Zaman, for your valuable time and great insights related to the development of the brain. We have 4 questions here. If you allow me, shall I move on with that?
Yeah, sure.
Thank you so much. Question number 1 is, can these organoids be used to treat degenerative disorders?
Well, these organoids, as I said, they can be used to model the as I have shown, they can be used to model neurodegenerative diseases. And then one can study the effect of various interventions. For example, even in the study that I have shown, what they did was that in the Alzheimer's disease model, they use this isogenic model that they, in place of expression of ApoE4, they had this, they switched to the expression of ApoE3, and that showed a decrease in the plaque formation and a decrease in the phosphorylated tau. So yeah, they can be used not only to model the disease, but also to study the effects of various interventions, be those the gene editing technologies, or be those the newer molecules that can be used to study neurodegeneration.
Okay, all right. Second one is, what are the chances of success if these brain organoids are inserted in human brain?
Well, it's a very interesting question that can these brain organoids be used? Can they be transplanted in the human brain, and then they function properly? Well, we are actually moving towards that. The research is moving in that direction. So like one of the ideas is, for example, in Parkinsonism, in Parkinson's disease, you can actually grow brain organoids that that have these dopaminergic neurons, and then the idea is actually to use those brain organoids to turn them into the functional brain organoids that can be integrated into the adult brain. So, well, the hope is that in near future that they can be actually used, be transplanted in the humans, and that's true for not only the brain organoids. That's like for, can we use liver organoids to like generate functional units of the liver, and then they can be used for the transplantation.
That seems really interesting. Another one is, can these humanized animal model be used for non-invasive study models?
Sorry, can you repeat the question?
Can the humanized animal model be used for non-invasive study model?
Yes, yeah, definitely. Like this humanized mouse model is already being used to study the like Alzheimer disease, and a number of other diseases. But I think what is interesting and what you are going to see in near future. I mean, there are a few groups that are working on that, but I think what you are going to see more and more in the future is that the mouse would be used as a model, but then uh, the brain organoids would be transplanted, human-derived brain organoids would be transplanted in the mouse brain, and then one would be able to study the interaction of these brain organoid cells with the microglia derived from, for example, the mouse. So that's these are the future dimensions of brain organoid research.
All right. So we have another question from Shuja Heather. He wanted to ask that, can we use this application for the dyslexic brain?
Okay, right. Yeah, so like if we are talking about hereditary forms of dyslexia or even the sporadic form of dyslexia, we need to understand that brain organoids can give us the a number of set of informations. For example, we can look at how dyslexic brain develops differentially from the normal brain, control brain. So we can look at the stem cell behavior in those brain organoids. We can also look at the RNA expression profile between the control and the patient-derived brain organoid model. So yeah, that can be used, but the limitation is that but what we cannot do is, unlike the animal model where you can actually see the dyslexic model, you can actually see the dyslexia. You cannot see, but that's a limitation with the brain organoids, obviously.
All right. Another question from Dr. Nazakat. She wants to ask that how to evaluate AD symptoms, or AD, how to evaluate AD in patients, basically.
Thank you for the question. I think that that's another detailed and extensive, and I think that's part of this program. Someone must be talking about, going to talk about the evaluation of patients with Alzheimer's disease. But well, the main feature of Alzheimer's disease is the dementia, which is loss of memory, inability to make new memories. And also there are some behavioral abnormalities that we see in the Alzheimer's disease patients. But I think that is the scope of another full talk.
All right. The last question is from Rijda Afroz. She wants to ask, what are the chances of contamination while working with organoids?
Yeah, so I think anyone who has worked with the IPSCs, they know that, I mean, with the cell culturing techniques, there is risk of contamination while working on these cells. So that's, I mean, you need to take the same precautions. You need to work in the clean working conditions while working on the brain organoids.
All right. We have one more question again. She wants to ask, can organoids be used for the treatment of strokes as well to translate them in places of neuronal death?
Well, I think the question is, one of the problem with the brain organoids, most of the methods that are being used is that brain organoids usually grow to a smaller size. It's not like the brain organoid is going to grow into a full-scale brain. That's not what we see. Um, and they remain in, I mean, they are of smaller size, and also they lack these blood vessels. So one might see hypoxia towards the center of these brain organoids. And so now people are working on the the improved models where brain and endothelial cells, and then we can study the effect of like increased or decreased blood flow to the brain organoids. But what I understand from here is like, can we use organoids as a source of neurons, and then we place them into patients with ischemic brain? Well, I think it might take a lot from now to get something like done. That requires a lot of experimentation, standardization, and to really see whether they are going to integrate into. It's not like just replacing the neurons. The important thing is that those neurons should make functional circuitry with the other neurons in the brain. Only then that's going to be beneficial.
All right, one of our participants wants to know about the NAD and SIRT activation in neurodegeneration.
Well, I think I don't know much about that because my area is not on the neuroregeneration. I primarily work on the neurodevelopmental diseases, and we will be using in the future these IPSCs to develop brain organoids and to study the neurodevelopmental disorders. Sorry, I think there might be other esteemed members in the panel, maybe later on, who can answer this question.
Sure, no issue, sir. Can you please give us some light on the role of exosomal therapy in neuroregeneration? Again, if it's in your context.
Yeah, so it's like it's more of the neuroregeneration, but I would say like exosomal therapy is again, it's very very interesting therapy. It's a very interesting way of delivery of different molecules to the cells of your target. I mean, there may be, I mean, there are emerging roles of this exosomal therapy in a number of diseases, but specifically neuroregeneration, no, I'm not sure.
Okay, all right. Thank you so very much, sir. We greatly enjoyed your talk and your session. Thank you for your valuable time.
Thank you very much.
Welcome. Moving ahead, we are not taking a break for this session today because of absence of our few speakers. So we have our last activity of the day. Let me introduce you all to the Dr. Meha Fatma from Dow University of Health Sciences, Karachi, Pakistan. Dr. Meha Fatma has obtained her PhD from International Center for Chemical and Biological Sciences, ICCBS, University of Karachi, Pakistan, followed by a postgraduate diploma in Mind Sciences from Preston University, Pakistan. Her PhD research focused on the development of a drug for the treatment of Alzheimer's disease patients with diabetes. Dr. Meha is currently serving as Assistant Professor at Dow Institute of Medical Technology, Dow University of Health Sciences, Pakistan. She has been working on cell culture, biomedical engineering, and neurosciences. For her recent research on the effectiveness of theta frequency sound in chronic stress, she has also received SRGP research grant from the Higher Education Commission, HEC, and Transformation International Society Research Support Fund. We welcome her on board with us. Greetings, Dr. Mia. Can you hear me, Dr. Neha Padma? Please wait 2 minutes while she joins us. In the meantime, Dr. Meha Fatma is joining us. Let us all enjoy the beats of the national anthem. Welcome back, everyone. Dr. Meha Fatma has joined us from Dow University of Health Sciences. We welcome her on board with us. Over to you, Dr. Meha Fatma. You can continue.
Assalamu alaikum and thank you very much. I just had minute problem with the internet And I've connected with the hotspot right now. I'm telling this because as soon as I get my internet connection back, I'll just connect it with the fast speed internet. So thank you so much for giving me a chance to learn from the participants and to share my meager amount of knowledge with them. Um, this time, the topic of our school or the workshop is the role of electrophysiology and brain mapping in neurodegenerative research. It's still under consideration and under rigorous research. The field is still developing because there always comes a question mark when we talk about using electroencephalography in the diagnosis of dementia. But if not clinically approved, but yes, it is very much um, an active field of research, an active field in the research side. And with the advent of advancements in technologies and different modes of analysis, with the advent of artificial intelligence and signal processing softwares, we are having better opportunities to identify the electrophysiological changes when it comes to neurodegeneration. So I would just start sharing my screen. I hope you guys are able to view my screen.
Yes, ma'am, it's visible.
Thank you. Uh, so, uh, Number one question that I used to ask myself, or anyone asks me, that if we have bedside examinations like MMSE and other, for example, 6CIT and other questionnaires available for differential diagnosis of Alzheimer's disease or dementia, then why do we need practices like electrophysiology or techniques like electrophysiology? We also have brain imaging techniques like functional MRI and SPECT scans. Uh, but they're very costly. It's not very common to use it in our setting. While electroencephalography or electrophysiological studies offer a better opportunity and a cost-effective opportunity when it comes to understanding the brain mechanisms. So this is, of course, number one reason. But apart from that, we have other reasons as well. Like why exactly? Uh, what we observe or how it has helped us in understanding neurodegeneration is that we have observed certain changes in the electrophysiological patterns in the patients who suffer from dementia or neurodegeneration. It also includes mild cognitive impairment like uh, the early stages, like in the early stages of Alzheimer's disease. Someone has just asked a question about how can we diagnose Alzheimer's disease. Yes, I can in part answer that question that we have some molecular markers as well, and now we are lucky to have some electrophysiological markers as well. So what we have observed so far when it comes to studying the electrophysiological patterns and neurodegeneration is that we have been observing a shift of power spectrum to lower frequencies. Um, because degeneration actually makes us lose our brain cells. So when we lose brain cells, the altogether activity inside the brain decreases, and that is the reason that when we plot it on the graph or on a pattern, and we take the pattern as a picture or an image, we observe a shift of power spectrum to the lower side frequencies like delta and theta. We'll discuss that or we'll mention it again when we will be talking about the patterns that we can observe in neurodegeneration. Um, Again, the other thing that we observe in electroencephalography for neurodegeneration is the decrease in coherence of fast rhythms. So, different brain areas, different regions of the brain work in connection with each other. So, if we have to recall a memory, it's not just the hippocampus, or if we have to make a decision, for example, a motor movement based on an event that, okay, let's suppose I touched a hot object and I remember that it was hot. So I won't be touching it again. Or if I touch it, I'll use a protective equipment before touching it. So it's all coordination between different areas like motor cortex, like hippocampus, like the cerebral cortex. And of course, different basal ganglia regions as well. They connect all together, they work in coordination, and then they produce a response. So when they are transmitting signals, of course, it is all electrical signals. So the electroencephalography equipment is now able to catch those signals and presented, and then we process the data to see if it is really coherent with each other or not. So what we have observed is that with the loss of neurons in neurodegenerative diseases, there is a decrease in the coherence of fast rhythms. And that is why the executive functioning, that is one of the predictive markers of, or one of the markers, the earlier markers of diagnosing dementia, that the executive functioning, the organizing capacity, the thought process, the connecting of dots between different things and making a decision ultimately, that ability is reduced in high cognitive impairment. So if now we look at the electroencephalographic patterns, we now know that there is a decrease in the coherence of those fast rhythms that helps to coordinate with different brain regions and then give a response. Then we know that we get mixed signals, and there is a terminology when it comes to electroencephalography, and that is EEG complexity, because the signal, we have these different signals at different time points, and when we are when we are recording the EEG signals, that complexity pattern changes in neurodegeneration. Um, yes, we can ask that what exactly are the changes, but there are a lot of them. And we need to have a basic background on how it works in order to discuss them in detail. And still, they are not in the diagnostic phase. But yes, they do play a role in predicting or in suggesting of what could be going wrong inside the brain when neurodegeneration has started. Um, And all these patterns, like the shift of power spectrum to lower frequencies, this decrease in coherence, this EEG complexity changes, the differences in the brain powers, they all are now hypothesized to be associated with the functional disconnections. So it is just like having the wires broken. So if we have the wires broken, we cannot join them again or fix them again when it comes to brain. So once it is lost, the flow of data from one wire to the other wire, the continuity of the transformation of the transfer of signals from that wire, that route is now broken, and that is why it is thought to be associated with the functional disconnections among the cortical areas uh, that result because of the death of neurons, axonal pathology, and the cholinergic deficits that result from axonal pathology. Because if we are losing the axons, we also lose the cholinergic transmission. So here I have indicated it through a picture that this is what is going on. There is axonal pathology, there is loss of cortical neurons, there is breakage in the synaptic connections. And how does it result? It results in certain features in the EEG. When we transfer this data from our recording software to let's say to any analysis software, for example, we use EEG lab for analysis of the data. So what we observe is that there is increasing delta theta ratio at the posterior temporal and the prefrontal cortex in the MCI patients. Also, we observe a decreased alpha wave at the central region, and here we can observe the central region, the C3, C7, and C4 electrodes. I'll be showing how do we actually measure that electrical activity from the electrodes. But the CZ or C just represents the central region. So we observe decreased alpha wave at the central regions, and also we observe increased beta activity in the temporal region. These markers have been studied with reference to uh, uh, discriminating vascular dementia from Alzheimer's disease and identifying semantic dementia as well. So initially, I remember almost 10 years ago or 11 years ago, I was in a conference and I had discussed to a clinician that I'm actually trying to identify if electroencephalography can be used for dementia. And he immediately responded that it's not specific, so we cannot have any definitive markers. And I couldn't believe myself when I was preparing this that after 11 years, I've now come to know that the accuracy and efficacy of electroencephalography has improved to the extent that we have now identified certain patterns. Of course, it is not still not definitive; it is still growing. But the good news is that it is growing. For example, initially, the cases which were correctly identified were between 29 to 42%, and with increasing technological advancements and better signal processing opportunities using softwares like MATLAB or EEG Lab, uh, it improved to almost 60% to 80%. And now we are at, at a stage where we are also developing some exercises that we count under the umbrella of neurofeedback. And these exercises do improve attention, memory, or at least executive functioning that are characteristically lost in or reduced in mild cognitive impairment. So, because it's a workshop, so I'll be discussing the basics of EEG, and I will be sharing a demonstration, and then I will be giving a demonstration as well. So the basic setup that we need is electrode cap and here, and then we need a conductive gel for that. We need syringes where we can fill the syringe with the gel. We need amplifier that will connect my electrodes or electrode cap to my software that will be recording the data. And there is a specific room setup. Ideally, there has to be a see-through window, and it has to be a separate room where there is no noise. It is magnetically shielded. Because I work on sound-based therapies, so I prefer a soundproof room. And right now I'll be demonstrating this data collection over here, right here, because I need a better internet connection. But we ideally use a separate room that is distinct and that is soundproof, so that no external sounds can interfere with the sound differences that we are working on. So moving on, how does it work? The electrode placement. There is a certain way that we place our electrodes. There are different montages, and what we focus is that every region in the brain is correctly identified or represented when we are collecting the data. So we place the cap in a way that different regions of the brain, for example, frontal is represented by the frontal lobe and the frontal electrode, and they are placed at a certain distance. Now, that system, where we have identified a certain distance between different electrodes, is called the International 1020 system. And what happens in the International 1020 systems is that we have this initial distance of 10%, and then onwards, as we move onwards to the scalp, there is a 20% increment in the distance of one electrode to the other electrode. This is especially crucial when we are using external or free electrodes. Now, luckily, because we have cap, so they are placed in a way that the initial distance is 10%, and the rest of the electrodes are distanced at the 20%. Of the distance from the previous electrode. So the cap already has this placement, and when we place the cap, we make sure we take the measurement from a point, which is this bridge, the nose bridge, and we call it mesion. And there is a bony protuberance, which is called inion. and we take the measurement, we divide it in the half and obtain a vertex point, because at this vertex point we place the central Z electrode, the CZ electrode. What Z means, I'll be sharing it soon. So the other measurement that we take is from one preauricular point to the other preauricular point. So we take the measurements from this preauricular point to this preauricular point, and we divide it by 2 to obtain the central measurement, like whatever is the measurement on the central side. So from nasion to inion, and between the two auricular points, we obtain a vertex point, and that is the place where we place our central electrode. Uh, so usually the placement is in such a way that odd numbers of the electrodes, for example, F3, FP1, C3, C5, all the odd numbered electrodes are placed on my left side or on the participant's left side, and all the even number electrodes are placed on the right side on the skull of the participant. And the midline, there are electrodes on the midline area, and they are represented by Z, which means 0, because there is no angle over there. So the odd numbers are placed on the left side, and the even numbers are placed on the right side of the participant's head, and the central electrodes are placed in the central region, represented by CZ. And this is the ultimate picture that we get in cross section, or in a 2 dimension. If we are going to observe the participant, we will see that all the electrodes, odd numbered electrodes, are placed on the left side, the even numbers on the right side, and the central region. So this is the sagittal plane, this is the parasagittal plane, and this is near the preauricular points, or away from the midline. So the lower numbers, for example, 3 or 4, they are placed in the parasagittal plane closer to midline. For example, this FZ, CZ, and PZ region. And the higher numbers are placed somewhat away from the central midline. And this is how the ultimate pattern of the cap appears when we place the cap on the participant. Okay, so what is the basic principle through which we record the EEG? It follows, or it provides us the signal based on the principle of differential amplifier. So how does differential amplifier works? It takes a positive reading, it takes a negative reading, it cancels out all the common noises or the common signals and presents the differences. For example, right now, it may be difficult if we are having people from background of biology, being a student of biology. I know it's very difficult to understand the concepts of physics. So I'll be giving a very common and easy example for that. For example, if you see this picture, there's a very common picture that we have, and we play it like identify the differences. So now, if we identify the difference, your brain will automatically detect that this portion is different in this picture, and it is different from the wall, and there is sun and there is no sun, and there are clouds, there are 5 fishes and there are 4 fishes, and etc. So your brain will specifically target only these regions which are different, and it will give you a perception of the differences only, because your brain has amplified this information in a differential manner. So it is only giving you the information about the differences. You're focusing only on the differences. So similarly, the brain gets these signals from one electrode and the other electrode, and then it amplifies the signal where it is different, and it cancels out the common signals. Um, then there is a concept of common mode rejection ratio, which is pretty much similar to the concept of differential amplifiers, just the difference. There's a minute difference, which is actually, it incorporates 2 characteristics of electroencephalogram or the electroencephalography. Number one, it is actually the ability of a differential amplifier to eliminate or attenuate common mode voltages, which are represented by VCM. The voltage that is constant for both the positive and the negative inputs of the amplifiers for the common signals. And it also requires its capacity to amplify the differential mode voltage, which is represented by VDM, or the voltage difference between the positive and the negative inputs. So it also is the capacity of attenuating the common mode voltage and amplifying the differential mode voltage, which means its capacity to identify the differences and amplify that signal, and its capacity to uh, identify the the differential mode and to highlight that signal. Okay, then the sampling rate. We have to be very specific about the sampling rate when we are collecting the data, because it is one of the main things that we use when we are analyzing the data. So the sampling rate is actually the number of signals obtained in a given unit or measured at a given unit of time. For example, we usually use seconds to obtain the rate when we are talking about electroencephalography. So it has to be somewhere between uh, 0.5 and 80. The generally frequency to be measured lies between 0.5 to 80 hertz, and then we measure it per second or per unit measurement, that is usually in seconds when we are recording electroencephalography data. The sampling rate has to be at least twice the frequency of interest or the frequency to be measured. So yes, if we say that at least 160 is required, that will be true, because it is opposite to the 80 hertz. That is the maximum frequency that we need to measure or that we usually obtain in an electroencephalogram. Ideally, for neuroimaging research and for research purposes, we require an equipment which has the ability to give a sampling rate of at least 256 hertz in order to provide a better resolution when we transfer our signals into or when we are going for the analysis of our signals. And here I have shown also the bands. For example, let's start from delta. So from the 0.5 hertz to 4 hertz frequency is of the delta wave. From 4 to 8 hertz is of the theta wave. From 8 to 12 hertz is of the alpha wave. There is a frequency between 12 to 15, and that that was initially discovered in cats, and it was characterized in sensory motor rhythm (SMR), and in humans we call it neurons. So that is why you see 12 to 15 hertz missing over here. And then from 16 to 24 hertz, there is beta. And then there are also frequencies beyond 24 hertz up to 80 hertz, and they are categorized as gamma waves. And they're mostly representative of the eureka movement, like when you are stuck somewhere and your brain is working really fast, and then you suddenly come up with a solution of a problem. Okay, so the montages that we use, how basically when we are having a representation of the data on the software, uh, the data is shown based on a certain placement or a certain measurement that we are obtaining from the electrodes. So if we are talking about montages, we do have bipolar montage and common average reference montage. For the bipolar montage, what happens is, for example, that this reading from FP2 to FP8 is obtained, and then the difference of FP2 to F8 is obtained, and this is how it is represented. Let's suppose from FP2 to FP8, this is the right region or the right channel. This is categorized as channels. So from the right channel, we can have this FP2 to FP8 readings. So this is using two electrodes and identifying the difference between the two electrodes and then obtaining the data. So we represent it on the graph as FP2 to FP8, and and this is typically a bipolar montage. We can also use it by providing the difference with the reference, with the average of all the electrodes that we are using. For example, if not this, in this picture, we are using an average of FB1, FB, F3, C3, B3, and O1, and all of them are being averaged, and the average is now differentiated from the electrode of interest. And when we obtain a graph, you will see the next electrode. For example, here in this F7, it is replaced by the word ABG, which reflects average. So we can represent data, or we can obtain data either through a bipolar montage or through a common average reference montage. Uh, Once we have EEG, what we are expected to look for in it, especially the artifacts, because these artifacts do hinder data. I have repeated it several times in the video where I have shown the demonstration as well. And even I will repeat it now, that this was one of the key factors that I learned when I initiated learning electroencephalography. That was, there is no substitute to good data. So we ensure that artifacts are minimized. There are a lot of sources of artifacts. For example, the power lines, the thermal radio frequency noises, eye blinks, jaw clenches. Most of the time when I'm collecting data, the participants' eyes are closed, but they are having this salivary production, and when they engulf that saliva, that also produces some kind of artifacts. And then even if they are moving their heads like from inside, it also produces artifacts. So and then there are certain electrical gadgets that may also produce artifacts. So we need to make sure that we, even the sounds, one of the most challenging aspects when I collect data is removing the sound of interest, or discriminating the sound of interest from other sounds. So they may cause artifacts in the electroencephalogram. Then we need to identify the normal patterns like the theta waves. I've already discussed the frequencies previously, the delta waves, the alpha waves, and the beta waves. Also, now there are some researches that are focused on gamma waves, because the gamma to theta ratio also provides a prognostic value that I'll be mentioning in the coming slide. Usually, when we obtain data from a scalp, the voltage usually falls between 1 microvolt to 100 microvolts. If we provide a direct intracranial recording, if we obtain a direct intracranial recording, we may obtain data in millivolts. And the reason behind that is the impedance that occurs due to the skin and the scalp and the skull. So, because we are indirectly measuring it, the amount of voltage that we obtain in the electrodes is in microvolts, and that usually falls between 1 to 100 microvolts. Why is that important? Because when we are removing artifacts, when we are processing data and we are removing artifacts, what we usually do is that we keep 100 as the cut point, and everything that is beyond 100, we remove that from the EEG signal. Usually, we make right to left comparisons and front to back comparisons, especially when we are working on neurofeedback, and we have to plan protocols, and we have to define the differences in different diseases. So I'll just share what usual brain pattern is in right to left comparisons and front to back comparisons. So in right to left comparisons, there is more alpha on the right side, on the right brain, and as compared to the left brain. On the left side, we have more beta as compared to the right side. Remember, this is the relative thing that I'm talking about. And then theta is more or less similar in both the hemispheres. When it comes to front to back comparisons, the alpha is usually higher on the occipital side because it also has the visual cortex. So we observe a very high alpha value when we ask the participant to close the eyes, because the signal to the occipital side or to the visual cortex is shut down. So we usually have higher alpha on the occipital region as compared to the frontal region. We have more beta on the frontal region as compared to the back side of the brain, because we are more into making decisions and executive functioning. we do that with the help of the frontal lobe. And theta is more on the frontal side as compared to the back side of the brain. And the general functions of the right hemisphere usually include the synthesis, social cues, emotional expressions, art, and language, even though we have Broca's and Wernicke's area both designated for language and language perception and understanding. But then also for music, for the perception of words, emotions, that is predominantly processed by the right side. It does not mean that there is nothing on the left side. Of course, the left brain is also coordinated, but we are talking relatively, that relative to left brain for the things like social cues, emotion expressions. The right side works predominantly, and for the rational stuff and for the logical stuff like mathematical calculations, logic, analysis, the left side is more dominant. So it works predominantly relative to the right side. So this is what we usually obtain in a normal electroencephalogram. But what have we observed so far in neurodegeneration? We have observed increased widespread delta and theta activity. So overall activity in the delta and theta region is more. Posterior alpha is reduced, and posterior beta is also reduced. Um, we have observed disrupted functional connectivity between frontoparietal regions and the frontotemporal regions, and this is how we were able to discriminate between the vascular dementia and cementing dementia. Then increased omega band complexity and lower synchronization between different parts of the brain. Also, electroencephalography has been researched for its prognostic value. There is increased theta to gamma ratio that suggests conversion of mild cognitive impairment to Alzheimer's disease, and also increase in the high alpha frequency has shown association, or it is now able to determine that this MCI patient may lead to Alzheimer's disease in future. So, electroencephalography is also now being considered for obtaining the prognostic value of a patient with MCI. So now I would just go with the tutorial. I have a pre-recorded tutorial from a known MCI patient, and I will be showing, I'll be sharing that video. I haven't removed any bloopers or any mistakes from that recording, because I think we are all learners. At least I categorize myself as a learner. So I would just show it as it is, and also the analysis part, how I have analyzed the data. If you think that we're running short of time and you would like to have a um, live demonstration, I would be happy to share a live demonstration instead. So here we go.
All right, everyone. Sorry for the little pause here. Dr. Mia Fatma is away for a, she's here with her live demonstration. It would be great if you please unmute your mic as well, Dr. Mia Fatma.
Okay, that is because I just muted the mic. That is why, Dr. Deepak, that was the reason you were not able to hear it. Now I'm just starting my video sharing. Okay, so I will first of all thank the participant for consent to participate in this video demonstration. I just have all the equipment that I just discussed in my presentation. This is the amplifier. I have this electrode cap and the connector, and I have these channels with me. I will just first provide demonstration of how to place the cap on the participant's head, and I will later on connect it to the software, because once I connect it with the software, I will not be able to show the video. I'll be sharing the screen only. So for now, as we have already shown that we need to have a central point or the vertex point where we place the central electrode. So I have this cap and it is tagged already with the different electrodes. Let me zoom in a little bit so that you can have this idea of how these electrodes are tagged. We have this FP1 and FP2, and similarly we have this central region FZ and the CZ, the most important electrode that we have to place on the vertex. So prior to this demonstration, I requested the participant again to have measurements, because I had to lend my measuring tape to someone. So I initially did the marking, and now I have this marking placed over here at the CZ region. I will now place my cap, and we have certain electrodes for references as well. Take them out. Before placing the cap, I preferably go for my reference electrodes placed on the preauricular region. Mostly, I place it near to the preauricular region on the earlobe as compared to the preauricular region itself. There are certain caps that provide reference electrodes for the preauricular regions. So I'll take the gel and I'll attach my electrode on this earlobe. and the other one on the other earlobe. Now I will place the cap and making sure that my central region, the CZ electrode, is placed exactly on the region where I have marked the vertex of the scalp. And as I have mentioned earlier that we have different electrode size, our participant is serving as a healthy age-matched control, and the video I have recorded is for a known case of mild cognitive impairment. Before inserting the electrodes, I have to make sure that they are not dry, and for that I will be using my conductive gel. Also, the electrode stickers are used for placing the ground electrode on the back side of the head. The back side of the neck. That is where I have placed the ground electrode. And I will first insert the gel inside the electrode. Usually when we have participants with more hairs, we have to take some time to ensure that the gel reaches the scalp, because if the conductive gel does not reach the scalp, we are going to have a lot of noise. I have a montage defined for the prefrontal regions, for the frontal regions, for the central region, and for the parietal lobe. I will ask the participant if he is able to feel the gel. Yes. Thank you. So yes, our participant is able to feel the gel. That will ensure us that the gel has reached the scalp. Also, one thing that we have to make sure when we are collecting data or we are using electroencephalogram is that we need to have our electrodes washed and clean, because if the electrodes or the gel crystallizes inside the electrodes, it will be difficult to obtain a clean data. Okay, so the way we place electrodes can be different for different equipments. They do provide you demonstration on how can you use their equipment for placing cap. This is the kind of electrodes that I have. So for example, my active electrodes are represented with red as I have shown previously, and I have this ground electrode presented in black. And I'll place all my reference electrodes on the side where I'm placing my red electrode. So if I'm placing number 1 on the left side, I'll also place its reference number 1 electrode on the reference region where I have placed my electrode. Right now in this demonstration, we are likely to have certain noises because I have some gadgets attached here. I'll ensure to minimize the noise, but since it is not electrically shielded room, so we might have some kind of noises only for the demonstration purposes. When we will be using the original, when we will be collecting the original data, we'll ensure a separate room where there is a see-through window. Now I'm placing my reference electrode. I'll place my active and reference electrodes in a similar way as I have placed them on the left side. I'm going to place them on the right side of the head. I'll join my ground electrodes together to place them on the back side of the neck. Right now, I'm attaching the reference electrode. Here, I do join my ground electrodes, the one colored with black. And I will placing one of the electrodes to my electrode sticker. And here I've connected the whole cap and electrodes to the head of the participant. And in order to secure this, we can tie the cap. So that our central point remains at the vertex where we have marked the central point. I'm now connecting all these wires or my electrodes to the amplifier, and here I connect my amplifier with my computer. The equipment that I use offers two options. I can connect it either through Wi-Fi or Bluetooth or through wire. Now you have seen the electrode placement. I will now open the software. I'll just close the video for now and start screen sharing, and will open the software and will show you how to connect it to the software. Depending on the software and your hardware or the company from which you have obtained, there can be a variety of options. I have designed this protocol. I can also collect data only from 4 channels or from 2 channels or from 6 channels. But for this workshop, I have designed it in a customized way, and the software I use offers this option, but you can have other options as well. So initially, I would just connect my device. It does have my device connected, but every time I'm going to connect it, I'll make a search. I'll do a quick search. All over again, and it will search my device, and then again here is my device number, and every time it searches and shows this, I will connect it to my software. Now I've selected it. We place the user details and also details from the person who's collecting the data. I can give any personalized ID. Let's suppose I can give this one. And I will add the client. And this client has already been selected. This was the previous client who was a known case of mild cognitive impairment. And initially, I will start a run without saving my data. And again, you can see a lot of noise on the electrodes that are placed on the left side. FP1, F3, C3, and P3 regions, the ones that are connected on the left side. It is because these electrodes are used multiple times as compared to the ones used on the right side. And we are also having frequent disconnections and noise because of the reason that I have electrical gadgets attached nearby. I'll try to minimize this noise by removing other items anyhow, because I have two computers connected here. So you are able to observe the noise. Another way we can try to remove this noise is to insert more gel and ensure that the electrodes are placed in a right way. Why you are able to see this? Because I have used my finger on the electrode and you are able to see the muscular movements from my own fingers. Also, you can try to fix the cap to make sure that there is less amount of noise in the data. Please note that I have not started recording it yet or saving the data. I'm just watching if multiple manipulations can remove the noise from my participants' data. At times, it may take a while for the participant to relax. And after a 2 to 3 minute recording, you can have a smooth EEG signal. Okay, and again, we are observing disconnection. And that is because I have to place the Wi-Fi device near me. It's very common to have these kinds of artifacts or problems during data collection because we are not in an isolated room. Usually, when we are in an isolated space, uh, we have the opportunity to minimize voice and we remove all the electrical items, all the electromagnetic gadgets that are there. So we minimize the external noise. Right now, we are not able to do it. So there is pretty much of a possibility to obtain noise in the data. And I'll again select the user. have disconnected my device and I'm connecting it again. Okay, there was a similar problem when I recorded this data at a home-based setting. There was a similar reason. Uh, so what I will do right now is that I will be playing the demonstration of how can we collect data using the software based on the video that I have made previously. And then I will record the participants recording and will share the readings with you and will request you to actually make a comparison based on the manual calculations. So here I have this data. Okay, right now we have started recording the data. I use the IBRO software and I have different options of creating different protocols to search protocols. That I have already placed the data, I have already placed the protocol over here. This may vary from software to software, and you may see that probably the software or the equipment you are using have a different associated. For example, also in these other, for example, C3 or the C3 region that we are having. Now you can see this immense depth. This is a typical ionic artifact. We have to make sure that while we are collecting data and as well as important aspects of the data, that is, there is no substitute to good data. Yes, we can process it with MATLAB and we can remove noises, so now I'm going to stop this recording. I previously I think the user, and I'm unsure again how did we add the user. The number or any number that you have to be. And now I have the username. And now I can I'll show you how to analyze EEG data. For now, what you are going to do is that you have two values of different bands or different bands. For example, if we have XP1 and XP3, so we will be getting that. So I'll go to the analysis mode and after quantify and then what happens to be avoid some kind of articles because the patient there has their patient has to what happens. Usually, as I have told you that the range of voltage inside the brain lies from 1 microvolt to 100 microvolts when measured on the scan. So that is why I am using the 50 microvolt artifact. So this channel is free of kind of artifacts that I'm going to have at 50 microvolt. So it is not make much difference. So I will just remove off and analysis. So I'm not including that right now. Right now we're just computing the values from here. Mainly showing the screenshot so that you can have the readings and you can later the delta value to the delta value or the F3 region. Okay, so we have the readings from participant right now, and I would, if you want, I can share the screenshot, or if you would like to have these readings for now, you can actually note them down. Let me know if you would like to have the readings right now, so I should keep the video over here, paused over here. Thank you very much. You will be provided with the screenshot of the results by the organizers. For now, I'm again going back to connect my software and to show readings again from the healthy age matched control that I've requested to volunteer for this demonstration. I hope you're able to view my screen right now and the data is comparatively more clean. I'll just get a recording of 2 to 3 minutes so that you can have a recording for your convenience and your comparison. I'm going to stop it and I'll add the user. Save, I will enable saving this recording. Right now. I will request the participant to blink his eyes. Can you please blink your eyes several times? And you can see a dip in this data. You are again observing artifacts because I have my Wi-Fi and my PC connected near. So there is a chance that you are able over here, but you can still dip in the signal. I would now request the participant to clench his jaw so that you can observe a jaw clench artifact. Here you can observe jaw clench artifacts. Now sit relaxed. I will now stop this recording and we'll go to the analysis mode. It will take a while to load this data, as I have shown previously. Okay, so I can try removing the artifacts. From each channel. And as I remove the artifacts, you can see a change in the values. There are different reasons of why certain artifacts can work better on 50 microvolt removal, and this is because of the options that the software offers me. So I have the option to remove it either from the 50 microvolts or the 100 microvolts, depending on the amount of noise that is present or the amount of artifacts that are present in the data. Okay. I would again request the organizers to please take a screenshot of these readings for the participants. And please let me know if you have taken the screenshot or you have the data, so that I can request my participant to leave if he wants. Okay, thank you so much for the cooperation, and I would also thank my participant for volunteering for this demonstration. Uh, Now, I would request the participants to just compare this data uh, from FP1, F3, C3, and P3 regions. Please get the average of what are the absolute alpha values and the delta values, and similarly obtain these values from the FP2, F3, from the FP2, F4, C4, and P4 regions, and compare. And please do a right to left comparison. Meanwhile, I will say goodbye to the participant as he has other commitments as well. While you do the uh, uh, comparisons or manual calculations for both the previous data for the MCI patient and for our healthy control. Meanwhile, we can take questions if the participants have any questions. And once we have the results from the participants as they calculate or do the comparisons, the front to back and the right to left comparisons, um, we can have for that during that time we can have questions if participants have any questions.
Yes, ma'am, we do have from the participants.
Sure.
Okay. Number one question is, what kind of changes do we observe in EEG recording in patients of dementia?
There are certain changes that have been reported as of yet. Number one is the generalized slowing of the EEG frequencies. For example, we observe that there is more delta and theta activity, which are low frequency waves. And there, as we have discussed, that there is a front to back and left to right weightage of or the distribution of these bands or their values. But as we observe this EEG in a patient with dementia, we observe that the balance from right to left between the front to back values of different bands is also altered. So there is generalized slowing of EEG bands. And then apart from generalized slowing, there are also certain ratios, for example, theta to alpha ratio, and they are also indicative, they're altered, they are altered in dementia. Moreover, there is a problem with cortical slowing or the cortical processing of these bands. So that is one finding. And with other research papers, we have a difference of alpha and beta activity. So that is altered. It is also evident when it comes to right and left comparisons in patients who are suffering from both dementia and depression. So we observe a difference of alpha activity because alpha is usually more on the right side. So we observe that the left side alpha is increased relative to the right side. So there are certain findings. As far as proper diagnosis is concerned, there is no diagnostic value is still established. But yes, cortical slowing of the general slowing of the spectrum is one finding. And then differences in the patterns of different frequency bands is the other finding in dementia. Sorry, Zamina, I cannot hear you. Can you please unmute and
Yeah, Our participants also wish to know some changes or effects can be seen in Alzheimer's disease and Parkinson's as well.
Uh, yes, when it comes to determination of specific ratios, for example, when we obtain the frequency bands, as you have you already have this data now, that how do we get the absolute values, then we obtain ratios. We have this alpha theta ratio, and then we have this gamma theta ratio. But when it comes to Alzheimer's specifically, there is increase in the gamma to theta ratio, and that is what makes it distinct from the Parkinson's, because in Parkinson's there are other findings. So no, I'm not able to find a definitive study which has defined the difference between the gamma theta ratio to the difference between someone who has a gamma theta ratio in Parkinson's or a gamma theta ratio in Alzheimer's. But yes, the distinct feature in Alzheimer's is the increased gamma theta ratio.
Okay, all right. Mr. Shujajar, Heather wants to know about the effect of the EEG for deep neuronal circuit recording.
Deep neuronal circuit recording. I mean, I'm not able to exactly get the question. Can you please rephrase it or repeat it again?
Sure. He wants to ask, is EEG is effective for deep neuronal circuit recording or not?
Yes, we do have intracranial recordings as well, but I think I have mentioned it previously or not, that because we are recording from the scalp, there is different amount of voltage that we get. But of course, there are intracranial recordings as well, and we do obtain intracranial recordings, but that is pretty much invasive. So yes, when it comes to recording electroencephalogram from the cortical regions, it is more effective and more reflective when we are comparing it to the deeper sides of the or the basal ganglia, for example, because EEG provides a better temporal resolution as compared to spatial resolution. We have imaging techniques which provide better spatial resolution, but EEG is good for calculating or measuring it in the real time, the time at which it is happening. So it has got a better temporal resolution. So if we are recording it non-invasively, and if you're asking in that perspective for the deep cortical regions, it is not as effective as neuroimaging. But then we now have softwares, and we now have processing softwares which can help us identify the coherence and connections between the different regions, and that are also indicative. So yes, it can be used for deep cortical regions, but as compared to neuroimaging techniques, the deeper regions are not do not provide a good resolution as compared to the neuroimaging techniques.
All right, ma'am. Also, can we detect any past deformality or seizures using the EEG?
Yes, we do have. Actually, EEG was initially and is still clinically used for diagnosing and identifying seizures, epileptiform seizures, and even creating or discriminating between different types of seizures. For example, absence seizures, for example, epileptiform seizures, for example, generalized tonic-clonic seizures. So yes, we do have this facility with the EEG to identify seizures and also to discriminate between various types of seizures.
That's great. Thank you so very much for your time and for all the efforts. We greatly enjoyed your talk and session, and I hope participants did enjoy too. Thank you again for your time.
Thank you. I just have one request. Participants can share their results or their findings. For example, the comparisons, the left to right comparisons in the MCI patient that I have indicated, and also in the healthy control. And if they have any queries, if they have anything specific related, specific to ask related to those results, they can always email me.
All right, that's great. Do you want to discriminate the result as well, Dr. Meha?
Right now, because it was a workshop, so I have left it for the participants, and I wanted to engage with them so that they can actually identify the differences. But yes, later on, I can for sure provide them the discrimination between results.
All right, great. It would be really great if you please drop your email address in the chat box so that everyone can see it and reach you out later on. Sure. Thank you so much.
Thank you.
All right, great. Dr. Neha Fatma has already dropped her email address in the chat box. All right, thank you again, once again, Dr. Neha Fatma, for your time. Moving ahead, we are done for the day. Hope you all have had a great time with us. Thanks to all the amazing speakers and for the great sessions. We are receiving the feedbacks from our participants and aspiring learners. There's great to know about all the feedback we are having. So for now, it's end of the day. Thank you so very much, everyone, for being a great listener, for being a great participant, and for the interactive session. We all greatly enjoyed it. Hope to see you tomorrow at the same timing with next amazing speakers and sessions. Hope you will learn with the same spirit just like you did today. Closing the day here. Thank you very much, everyone. See you tomorrow. Inshallah ta'ala. Allah Hafiz.